All right, so when we last left off, we talked about the 6 general types of signal transducers, and we're going to go over these. We're going to start with GPCRs, the G protein coupled receptors, and really go the most detail in those. First of all, there's multiple steps, and secondly, they are very common. Then we'll cover the RTKs, receptor tyrosine kinases, the RGCs, receptor guanylyl cyclases, the integrins, which are also called adhesion receptors, gated ion channels, and nuclear receptors. Now, those last two we don't cover in as much detail for a couple of reasons. One, gated ion channels we actually covered a little bit last semester. You also cover them usually ad nauseum in A&P classes. And nuclear receptors because there's just not as much information. Okay. Not as fun. All right, this is a figure from your book that I really like, and as I mentioned, I actually use this as the outline to go along because it has all 6 of the different types, and so we'll be covering that as we. Here we go. So the very first one is called the GPCRs, which stands for G protein coupled receptor, and then we're also over what a second messenger is, and I will apologize now. I learned them being called secondary messengers, so I will probably call them secondary messengers throughout the semester. But second messengers or secondary messengers, it doesn't. Hopefully you get the idea. Okay. If it's not first, it's second. So that's what happens next. Okay. So in order for something to be considered a GPCR, you must have at least 3 essential components. First up is you have to have an integral protein, okay? And what does it mean if it's integral? It doesn't mean it's just important. You know, remember that we had integral versus, what was the other? Peripheral. And what was an integral protein?
It's like inside.
It's actually in the membrane. Almost always, offhand I can't think of any exceptions, but almost always, at least for GPCRs, it's a 7-transmembered alpha helical protein, okay? And this is an enzyme. Okay. The second thing that a GPCR must have in order to become a GPCR cascade is an intracellular second messenger. And then finally, it has to have a G protein, which makes sense since it's a GPCR. G protein is just the acronym for guanosine nucleotide binding protein. What do you suppose a G protein does? It binds. What nucleotide? Which is G. Okay, so yeah, it binds G. Actually, it binds GTP and GDP. We'll see why that's important in a moment. First, we're going to go over just a little bit about second messengers. These are some examples. There are a lot of different types, but CAMP, cyclic AMP, is the one that we're going to go over in greatest detail right now. But you have to realize that some of the others use different things. But these are like the 4 B's. The CAMP, CGMP is also used by some. Okay. There's calcium, and it's a calcium ion. Just remember, it's a 2+, it's a divalent cation. There's diacylglycerol, which sometimes in some papers and stuff they just abbreviate as DAG, D-A-G. And then there's an inositol. Okay, so here usually we're talking about inositol triphosphate or IP3 or PIP2. Remember we call them sometimes the PIPs. At least I call them the PIPs. Sometimes they're abbreviated that way, PIP2. What is inositol? Does anyone know? The OL indicates alcohol. It's a type of sugar. Sugars, alcohol is a type of sugar. So there are lots of alcohols on inositol. Right. We're going to go in the greatest detail with CAMP. This is cyclic AMP. I call it CAMP. Some people call it CAMP. CAMP is easier to remember. Excuse me while I take a bite. I wanted to put up here a couple of different ways that it's shown and drawn. The one here on your left is probably the way that you see it most of the time, but it's not very realistic. Okay, but it's to show you, a lot of times whenever they draw DNA and RNA, you see this part, and I think that's the reason why it's shown that way. This is done, I think this is using ChemDraw, but it kind of gives you a better idea of the angles involved. But they're both CAM. Okay. Where does CAM come from? ATP. Yep. Okay. This just shows you an example of some of the different functionalities, or phenotypes, I should say, that cyclic AMP has an effect over. So inflammatory processes, platelet aggregation, dilation, contraction, all that kind of stuff. So now what we're going to do is we're going to look at a very specific GPCR. That way you can look at it and kind of go over, see how it works, okay, rather than just doing it in broad generalities. And so, and I don't think on the one that I uploaded, if it says that we're playing a game, we're not playing a game. That was back whenever, that was a different class. Okay, good, it doesn't. All right, so this is the beta-adrenergic receptor system, okay, which beta-adrenergic is fancy terms for epinephrine. So we're going to look at epinephrine, or adrenaline. And then just to, I think last semester we went over this term, but if not, the difference between an agonist and an antagonist is really important, biochemically and biomedically. So remember, an agonist looks like the original substrate, and it elicits the effect of that natural ligand. Whereas an antagonist, also structurally many times will look like it. It has the same type of pharmacophore, for those of you in medicinal chemistry. However, it does not elicit the same response. Usually we're talking about without eliciting any response. So it blocks, like an antihistamine. Antihistamines block what receptor? This is kind of like the category of dumb answers. Antihistamines block what receptor? Histamine. Right, so that's why it's called antihistamine. Okay. So this just shows you some examples. We have epinephrine, isoproterenol, and propranolol. Okay. One of them's an agonist, one's an antagonist. Those are relative KDs. So you can actually see where the antagonist binds much more tightly than the actual original ligand. Mm-hmm. And let's see, the agonist binds roughly 10-fold, or about 1,000-fold tighter for propranolol. So you may, if you've forgotten what KD meant, you may want to review that from last semester. Okay, here's the fun part. This is where the game used to be, where we had the different names. This is a cartoon. We're going to take it through step by step. What do you suppose, for number one, is the very first thing? If we have adrenaline, okay, I almost always use the exact same example. Does anyone remember, did I talk about the example that I used with adrenaline last semester? It's running from a bear. And then I always say that there's I always put a little disclaimer that if you're going to run from a bear, you run downhill, not run uphill. Because, yeah. Okay, but if you see that bear coming at you, what's the very first thing for your adrenal? The epinergic, the beta adrenergic receptor system doesn't happen. What must happen? Binding of epinephrine. Okay, or adrenaline if you want to call it adrenaline. That's the very first thing. Notice here, we have the extracellular portion, we have the membrane, I feel like being a white, and we have the cytosolic side. This is faith integration for today. So the adrenaline is much like Moses with the Holy Land. Okay, so if this is the Holy Land, adrenaline can see but cannot enter in. So that is a commonality that has to happen for GPCRs. It's an extracellular event. The binding is extracellular. The original signal does not enter into that Holy Land of the cytosol. We have Moses in the Holy Land a lot this semester. I have to take faith integration right, get it. Okay, so it binds on the outside side, the extracellular side of the protein, of the receptor. What do you suppose has to happen? Like, how does all of this know what's going on? That the adrenaline has bound here. The adrenaline binds, but how does the G protein here understand what's going on? That's not the liver, that's the G protein. Change in conformation has to occur. So it binds here, there's a change in conformation. That change in conformation is going to signal the event on the inside. Okay. So the way that they, that's why it said worth double. All right. So it changes the conformation. G proteins typically, in general, it matters if it has a GDP bound, D as in day storm, versus a GTP, T as in terrific. Okay, so typically, if it's GTP, it is turned on. Okay, think T, turned on, activated. If it's GDP bound, usually that means it's deactivated. And so what happens is this conformational change that occurs at the receptor causes its affinity for GDP or GTP to change. Okay, so it displaces that GDP with a GTP. All right. Oh, I don't want to cover up the next one. Uncover it. So, because it gives away an answer that I want you to think about. So, let's just review. Once again, because I'm going to go over this reviewing quite frequently. Yeah. We had adrenaline that came, you saw the bear. You ran downhill from the bear. And the reason why you run faster and faster and faster with fight or flight, the adrenaline binds here extracellularly, causes a conformational change. That conformational change signifies to the G protein that, you know what? We need to displace a GDP with a GTP. And so if you notice, now only part of it, not the entire thing, but part of it actually goes on to this thing. In this picture here, they abbreviate it as AC. That AC stands for, it's an enzyme, adenylyl cyclase. So for those of you who've carried over your list of enzyme families, it's from last semester, and if you haven't, you should. And then we now have a new family name, and that's called cyclases. Okay. What do you suppose cyclases do? They can either break down cyclic molecules or make them. Make and break. Okay, they're usually involved with making something or breaking something that's cyclic. Okay. So that's why I know there's lots of boxes here, but I'm okay, so whenever so I forgot to point this out, but the G protein typically has at least 3 subunits, alpha, beta, gamma. Alpha is the one that has a GDP or GTP that's bound to it. The beta gamma ones are usually regulatory. Okay, so whenever the GTP is bound, it moved over here to the adenylyl cyclase. Now something else is happening that I've marked there with the question marks. What that is it requires an additional GTP to GDP. If you notice that, see, that's where we get this displacement. A GTP displaces a GDP. So now we go over here to adenylyl cyclase. That's that white enzyme. Okay. In this, the binding of the G protein, the activated G protein to the adenylyl cyclase is going to activate it. Whoops. But before I get there, I didn't realize that I put the regulatory part in here. So keep that in mind. Okay, let's see, can I make this a little bit smaller? Yeah, oh, awesome. So, this is just like a breakdown of that little portion that we've been talking about of the G protein becoming activated. Okay. And versus it becoming activated as just the alpha. So if you notice, we have alpha, beta, gamma. And so what happens here is anytime that the GDP is bound, it's turned off. So therefore it cannot activate adenylyl cyclase. What happens though is do that conformational change, then it's going to make it to where the GTP can displace that GDP.
And so then these subunits separate. This will get recycled back to be the regulatory unit for later on. This goes on, okay, and will activate adenylyl cyclase. Okay. Now, what's going to happen here is these G proteins have what they call the intrinsic GTPase activity. See, it's written down here. Okay, that's just an easy way. What does ACE stand for? Enzyme. And this is the enzymatic activity, and it literally means that it's going to break the GTP. It's going to take off a phosphate from it. Okay? And so it's going to hydrolyze it, and it's going to turn it to GDP. So they call it GTPase activity. Just like there are ATPases that are specific for ATP. But by doing that, now it's turned off, and it will go back and bind the beta gamma subunits, and we'll be awaiting for the next adrenaline. That's if you're healthy, under normal circumstances. And remember, just as a little review, this PI stands for inorganic phosphate. So, one way to tie this into medicine is the fact that this is the cholera toxin. So this is one way that cholera infects it. So normally we have this GTPase activity, right? And so the GTPase activity of the G protein itself will cause it to turn itself off after it's being used and go back and wait for the next adrenaline. However, the cholera toxin blocks that ability. Okay? And by blocking that ability to ever be turned off, it's going to constantly turn on adenylyl cyclase. So when you have the cholera toxin there, the GPCR system, the entire system, is constantly turned on, which would be bad. You have to remember, if you think back to SADI, you have to have some way of desensitizing, turning it off. Because otherwise it's just constantly on the I, or amplification. It's constantly on the amplification and integration for that matter.
Beta and gamma?
I don't know that it would. Just because if there's a lot of those receptors in the same area, how would it know? Okay.
Does it always go back to the same?
All right. Like I said, some of these little questions are the fact that we broke up his, back when I could give extra credit, we broke up the class into, I broke up the class into teams, and they're competing with each other. And then the teams that won got extra credit on the quiz. I did not vote in favor of the abolition of extra credit. Okay. So the enzyme is the adenylyl cyclase. The enzyme is the adenylyl cyclase. And I'm going to actually show its action on in the next slide. But if you think about it, adenylyl cyclase is going to take ATP and ultimately turn it into what? Adenylyl cyclase. C, not C-ATP. Camp. Remember, that's why I always say camp. That way you can remember it's only got one phosphate. Camp. Okay. And so this is a breakdown for all of this. Okay. Okay, give you a moment just to look at it, to take it all in, to absorb it.
Are you able to tell us what's behind the boxes?
Yes, but I'm giving you a moment to look at it.
I just want to make sure you
Should we be drawing it? You may want to, you could always write it out in just a moment. First of all, what is the name of that compound? What is that? Someone said it. Say it with conviction.
Adenosine triphosphate.
Yes, it's ATP. Adenosine triphosphate. Okay. I don't make you guys know how to draw the adenosine part, but you should recognize from the fact that it's got, it's like from enzymology last hour. 1, 2, 3 phosphates. Sorry, we're doing a lot with like that kind of stuff in our household now. All right, so that's ATP. So what enzyme? Don't overthink this. Oh well, actually, So what is this? That's ATP. What is the second molecule here?
Camp.
That's CAMP. just the way that they like to draw it. Okay, so that would be this box. Don't overthink it. Oh, and does anyone remember from last semester what PPI stands for? The I is inorganic and PP? 2-phosphate. It stands for pyrophosphate. That's what they call it. That's what they named it. Yeah, it's inorganic pyrophosphate. Okay, so what enzyme is it that goes from ATP to CAMP and gives off an inorganic pyrophosphate? It is? So let's review. What class of enzymes would be doing this?
Cyclic.
It's a cyclase. So what cyclase is this one? Someone said it, I think. Yes, the adenylyl or the A, the ATP, cyclase. That's what they abbreviate as AC. Adenylyl, because it's going to work on. Technically, there are two different molecules here, and they're both A's, right? So adenylyl cyclase. Okay. Now, as a little side note to think of in future, because I don't think that we cover it in this chapter, but inorganic phosphate is really an important compound in your body because it can be broken up into 2 inorganic phosphates by another enzyme called inorganic pyrophosphatase, and you get lots of energy from it. So it's actually one way that we can get energy. Little side note. Okay, let me show those boxes. So we have ATP, adenylyl cyclase, cyclic AMP, or CAMP. Oh, right. We have to have some way to break CAMP down, or otherwise bad things would happen too. Because remember, CAMP is a secondary messenger. It goes on to activate all the next enzyme, the next row of enzymes in the amplification process. So you've got to be able to break it down. And so this is what happens here. So we went from CAMP to this compound. What is this compound? AMP, AMP. It's no longer cyclic. Okay, let me make sure that I only do one box at a time. This one's not work double anymore, but it used to be work double. Okay, this is a very important enzyme, okay? Because this enzyme takes CAMP and it breaks it apart. So now CAMP no longer can go turn on the next round of enzymes. So this is the way that our body has in order to say, okay, we need the bear is gone. Don't keep on running. Don't be Forrest Gump and run, Forrest, run, kind of thing. No, I hate that movie. But yes, don't do that. So, I know that's like almost sacrilegious, isn't it? I don't get it. I don't get the love that people have for that movie. Okay, so now we have to think. What is actually happening here? We're taking the water, and water is being used as a nucleophile there to break this apart, alternately. Okay, so there is some type of a hydrolase. What else is there? There's another class of enzymes I don't think that we've actually talked about yet. In this class, you may talk about in genetics because it does have a role in that kind of thing. What else is there that we're doing here? What kind of compound is this compound in particular? It's cyclic. It's an ester. What kind of ester? Think about, how do we talk about nucleotides like DNA and RNA? What kind of backbone does it have?
Phosphodiester.
Phosphodiester. It's a phosphodiester. Okay. So this enzyme is a phosphodiesterase. It breaks it apart. Okay, and it's got a very specific name. I mean, it's cyclic nucleotide phosphodiesterase. That should give you a couple of hints about it. If I had just given you the name, literally what it does is it breaks down the phosphodiester bond of a cyclic nucleotide, and it also should tell you that it probably isn't too picky. It's probably a little bit more promiscuous, taking the CAMP or CGMP, or those are the two that I know that are biologically important. I don't know. I guess in theory, if something weird happened to not like CUMP or C, TMP or something like that, they probably would be able to have some sort of affinity for it. So this is one way, because AMP cannot do the same things that CAMP does. Our body's very specific. Okay, so let's go back to this one. So we've learned how to break it down. We have CAMP, when we don't need the camp anymore, we want to shut this off. This is where we're running from the Bayer. But we don't need to run from the Bayer anymore. We want to shut it off. We use cyclic nucleotide phosphodiesterase to do that. So now we're at this point. Camp has to go and do something. It's not just camp, just because you have camp there that you're going to be able to run. You know, and it's not just running. You know, you've got to make sure that you can, your blood pressure is relatively controlled. You're increasing your heart rate, all the stuff that adrenaline is going to do. You're going to downregulate the other body processes, increase metabolic breakdown of sugar and things like that, breakdown of fats. But camp by itself doesn't do that. So that means it has to do something else along this way. So does anyone know, what does camp activate? It activates a very specific protein. It activates pKa. Okay. pKa is one of the most generic named proteins that is there. All right, so we're going to break it down. The P stands for protein. The K, does anyone venture an educated guess what K stands for? Kinase A. This is protein kinase A. Okay, protein kinase A is turned on by cyclic AMP. A. The reason why I say that is there's also a PKB and a PKC. We'll cover those later on in the semester. So PKA, PKB, PKC. There may be more PKs out there. I'm PK. So, all right. So camp activates PKA. PKA is very promiscuous. It gets around. Okay, that's why it's called protein kinase. It has lots of substrates where it goes and it just starts phosphorylating other proteins. And those proteins, when they get phosphorylated, they get turned on or turned down. Turned on. No, activated or deactivated based off of their affinity. Okay, because for example, the enzymes that are involved for the breakdown of sugar would be getting upregulated typically, whereas for the synthesis of glycogen, it's going to be deactivated. Okay. I was going to go with that somewhere, but okay. I don't know what that is. Okay, but so pKa actually is a very complex allosteric unit, or it has multiple units. One of them is called ACAP, which is for A kinase incretin protein. But we have the catalytic subunit. We also have the regulatory subunit. What CAMP does is it displaces the catalytic subunit from the regulatory subunit. Okay, so until then the CAMP gets kicked off, then this would be active. And then the catalytic subunit is what goes on to do what pKa does. And what is it that pKa will do? Protein kinase A, what does it do? By virtue of its name. What's the protein kinase?
Phosphorus.
It phosphorylates
Proteins.
phosphorylates proteins. I heard someone whisper it.
Using ATP.
Using ATP or GTP. This one's ATP. Okay. And it's relatively promiscuous because it phosphorylates lots of proteins. Okay. That's why it's protein kinase A. It does have, like, its full official name is CAMP, cyclic AMP dependent protein kinase, but everyone just calls it pKa. Okay, and there is a PKB and a PKC, by the way, so we'll talk about those later on in the semester. All right. Actually, we already discussed that one. Okay, from the beta-adrenergic system. Yeah, so then pKa goes on and does the phosphorylation of a whole host, a bunch of proteins and enzymes, which will either get turned on or turned off based off of their regulation for phosphorylation. So you can imagine things that you need. You're going to turn on the metabolic processing for this specific receptor. We're going to turn on things that will help you make energy. Okay, going to release the sugars and break that stuff down. Break down the fats and stuff like that. You're going to down-regulate those that are going for the storage of sugar, the storage of fat, and all that kind of stuff. Because you're trying to run from the bear. Remember my little disclaimer: you always run downhill from the bear. Okay. All right. This just shows you examples. Okay. But you do not need to memorize this, but it should make sense why that these work. I could, and later on in the semester we're actually going to learn more about a lot of these, but I could, in theory,
at least ask you, so for example, on the histones, do you suppose that histone is going to be, upregulated or downregulated,
or explain like what you think would make sense for, in this instance, for adrenaline being released, are you going to want to do increase or decrease transcription? You kind of have to work your way backwards from that.
Okay. Some of these are much more obvious, I think much more obvious than others. Glycogen synthase. Synthases, that should be on your list of handy dandy enzymes. Synthases make, and so this make things, and this makes glycogen. You're not going to want to be making glycogen if you're running from the bear, so adrenaline is going to downregulate that. Okay, in this instance. Oh, actually, that's just the exact same figure that I had shown earlier. It's just blown up to show you some of the processes that are affected by CAMP.
I really like this figure a lot because it shows you how, it kind of uses algebra, but it shows you how a single, X number of molecules and the effect that it's going to have. So for example, if you just had one single binding event of epinephrine, you can see each different step along the way here. This just shows you for glucose, it's going to release 10,000 glucose molecules. Okay? So that's why it's called a cascade. It's going to the amplification of the signal that we already mentioned about the SADI. This is the amplification of that signal. So it's going to activate, adenylyl cyclates, which you can actually walk your way through a lot of this, which is going to ultimately activate pKa. pKa is actually going to activate phosphorylase B kinase. Just given this name, I would actually expect you to know what phosphorylase B kinase does. What does phosphorylase B kinase do? You should already know that, just based off of its name.
What cuts the phosphate? What?
So what does it do? Who wants to take it? Should I get to call somebody? It's like a little lottery. See, if I said I was going to give away bonus points, and people would be like, oh, okay, I'll do it all the time. So Matthew, why don't you take it? What do you suppose it's going to do? Phosphorylase B kinase. First of all, which end of the name should we start on? That's one reason why I wanted to pick this one. Do you start left to right or right to left? It's right to left. Okay, so we actually start here with a kinase. So what does kinase do? Phosphorylates using ATP or GTP, and what is it going to be phosphorylating? Phosphorylase B. Okay. Now I could, and the reason why is phosphorylase B is now going to go ahead and do, whatever it goes on to do. Okay, but so on and so forth. But yeah, that's why this is to show you how ultimately the goal is going to break down glycogen, make glucose 1-phosphate. Glucose 1-phosphate enters. Is there any what metabolic pathway this can go into now? In order to go to glucose, you're going to go through gluconeogenesis. And or at least a part, a little sidestep of it, in order to make glucose. Of course, then this gets released. But these are some of the other signals that use CAMP, the second messenger. Adrenaline is not the only one. Okay, some of these you may already know. Some of these we're going to actually go over this semester. We go over glucagon, but you probably already know. What is glucagon from anatomy? You don't. I see people get glucagon and glycogen mixed up. What's glucagon? Besides sounding like a monster from Battlestar Galactica. It's okay to laugh at really nerdy jokes. Okay, but what is glucagon? Perhaps some of you right now, your glucagon is really high. I don't know. Or low. What does it do? What does it help regulate? What is its enemy? If it's Lex Luthor, who is it? Superman? Insulin, thank you. Okay, so it's the opposite of insulin. And so insulin gets released for, when do you have insulin?
Blood sugar.
Right? Right, if you eat with high blood sugar, so this is for low blood sugar. It's the opposite. Okay, but yeah, a lot of these we already we'll cover, hopefully we have enough time to cover search on this somatostatin, and maybe a little bit of TSH, not FSH later on. Okay.
All right.
So these are some of the other ones that use different second messengers, and once again, this is just to show you that it's not only adrenaline using CAMP. You can use a lot of different things, and there are a lot of different
Aspects to it.
and we just use the one with CAMP and epinephrine as an example. Okay, this one is calcium. So we've already mentioned that calcium is very similar with GPCRs, except it doesn't do PKA. It activates PKC for protein kinase C because it's calcium dependent kinase. PKC. Does anyone want to venture guess what PKB does? What is activated with PKB? Is not boron. No. A does CAMP, C does calcium. What does B do? It's inositol. I have no earthly idea why they call it B. I think maybe it's because they had an A and a C and they thought, hey, let's have a B. But okay.
Instead of PKI or something like that. All right.